continuing on the metaplasticity theme, first of all, I want to thank Jennifer for the invitation. This is, as Rich was saying, is a fantastic meeting. It's great to be here. I'm putting up this slide to encourage you to come to New Zealand. It always looks like this, except when it looks like that, which it was about a week ago in Dunedin. So yeah, if you want to ski in August, you know where to go. Okay, so it's a slightly complicated title, but hopefully by the end you'll understand where I'm going. If we think about plasticity in the nervous system, there's plenty to go around. The toolkit is full of things that the nervous system uses for various purposes. We've talked quite a bit about a lot of these. A lot of focus on LTP and LTD, synaptic plasticity. But really, in my view, if you want to understand the neural mechanisms of learning and memory, you need to have a grand unifying theory that encompasses a lot of these things. So that's just my kind of little comment there about that, because I want to talk about another one, which is metaplasticity. And that term has been used quite a lot now, and I don't know how familiar people are, everyone in the room here, with what we're actually meaning by metaplasticity, so I thought I'd just go through that a little bit. So here I'm describing it as something like the plasticity of the neural state. I want to explain a little bit about what I mean. So this idea that there's basically prior neuroactivity produces some lasting change, you can think of it as a memory, that regulates the ability to get synaptic plasticity in the future. All right, and that may then also apply to learning. So experimentally, it's normally done like this, where you have some baseline period, and then you have some priming activity of some kind, or maybe you're reducing activity, and then you stop doing that, and then the cells are in a new state, a metaplastic state that could last from minutes to days or longer, such that when later on you give high frequency or low frequency stimulation, or you do learning, you get an altered synaptic plasticity or an altered learning experience as a result of the prior priming activity. And the priming activity might be neurotransmitters, it might be hormones, it might be various kinds of intercellular signals, and there's a whole family of metaplasticity phenomena, and you've heard about some of them already in this meeting. And there's various kinds of priming activities that we've been talking about. There's synaptic activity, you can control the postsynaptic cell firing, but you might have behavioral events like enriched environments or stress or learning that could serve as a way of setting up this metaplastic states and the relevant neurons that are involved, and then changing future plasticity. So what do I mean by state? Well, we know that synaptic plasticity is regulated by the instantaneous or the concurrent state of a whole bunch of factors that regulate synaptic plasticity. And again, you've heard about a lot of these already at this meeting. But for example, the exact complement of subunits in the NMDA receptors, the degree of cell excitability and the membrane biophysics that lead to a cell more or less able to fire action potentials, for example, the degree of synaptic inhibition from GABA, the presence of neurotransmitters, cytokines, hormones, modulatory neurotransmitters, they all regulate how much LTP or LTD you're going to get from a given protocol that you apply to the slices. So metaplasticity means that the recent history of activity can regulate these state variables and thereby influence plasticity. And there's many other variables that can be regulated, but these are just kind of examples. So the meta is meant to be something like the same vein as metacognition, meta-analysis, meta-learning, metaphysics, maybe. Not metaverse, we just kind of avoid that. Okay, now I mentioned that there's a variety of metaplasticity phenomena that have been described in the literature already. And so here's kind of a diagrammatic example. So one is that you could have altered activity at an input, and the state change is confined to the synapses that have been active. So we would call that homosynaptic metaplasticity or synapse-specific metaplasticity. But also there's examples where you have activity at inputs, and not only is the state change at those inputs, but maybe at nearby inputs as well. So it moves heterosynaptically into maybe the compartment, the dendritic compartment perhaps, where the change is occurring. Or you might have activity that drives a cell-wide change in some parameter that influences plasticity, regardless of where you're going to measure the plasticity on that cell. So that is more of a global state change as opposed to local state change. Now, I understand some of you are modelers, and people who do models, computational models of memory, frequently use metaplasticity principles in their models. So here's some examples of people who have used the principles either kind of consciously or unconsciously referring to metaplasticity or using metaplasticity principles in their models. Furthermore, I've discovered recently that deep neural networks also use metaplasticity principles particularly for the purpose of combating what's called catastrophic forgetting during the process of continual learning. So, you know, everyone's aware now of artificial intelligence, how wonderful it is, but it's a major issue is for networks is how do you retain old information when you're learning new information in that neural network that you're dealing with? Because if you don't control it, you can end up with what's called catastrophic forgetting. And for those of you who work in this area, if any of you might recognize the terms, for example, regularization, which is just another word for metaplasticity, basically, or changes in the learning rates in the networks or parts of the networks. So regardless of the biology, the concept has proven to be quite useful in modeling contexts. Now, coming back to computational models, there's been a lot of thought about, and I kind of referred to this in a way yesterday, that this idea of cell-wide metaplasticity might be needed for maintaining stability of the total network or total input of synaptic strengths onto a cell. And so the idea is, as I'll explain in a moment, is that you don't want inputs to become too strong, for example, and you might lead to hyperexcitability, or overall too weak. And so you need, so the models have incorporated cell-wide metaplasticity parameters to help with maintaining overall stability while the network or the cells are learning. So the classic example is something we've also been hearing about is the BCM model, going back quite some years ago now, gosh, 40 years. And it was actually, again, the physicists probably know who Leon Cooper is. He's a Nobel Prize winner with working on superconductors. Apparently all the work was done in his 20s. So, you know, I don't know what that says, but he decided to turn his attention to the nervous system. And so he was, so the group was looking at trying to explain plasticity in the developing visual cortex with the idea that if you monocularly occlude an eye, then the synapses, the visual synapses being driven from that eye become weakened. And yet when you do a reverse suture, those synapses can be stronger again, even though you might expect that they're too weak to undergo LTP. And so how does that happen? So he proposed, or they proposed that it's because the ability to produce LTP. So this graph here, for example, is how much plasticity you get depending on the size of the postsynaptic response for a given input level of activity. And if you have a high postsynaptic response, you get LTP, and if you have a moderate postsynaptic response, you get LTD. But their proposal, a key part of their proposal is, apart from that kind of learning curve, you might say, this crossover point between LTD and LTP can shift based on prior experience. So that if we have cells that have a lot of weak synapses, they don't lose their ability to show LTP because the threshold for LTP goes down. And indeed, in Mark Baer's lab, they showed, with Ben Philpott, they showed that if you do dark rearing and light compared to light rearing in rats, I think, you can get shifts of the kind of a BCM-like curve in plasticity, where LTP is easy to get in animals that have been dark-rooted. So two of the main postulates for the BCM model are this. First of all, as I was referring to before, it's a cell-wide plasticity shift. So it occurs on all the synapses, excitatory synapses at least, on the postsynaptic cells. So it's a heterosynaptic model. And the kind of the driver in a way as to whether to shift and for how much is a function of the time average degree of postsynaptic cell firing. So it's an action potential based theory. So this is my starting point, influenced by Mark Baer, turned out to be a bad influence, to try to understand the underpinnings of the BCM-like metaplasticity. So we moved to the hippocampus slice, trusty old preparation. And so we were studying here in the stratum radiatum area where the apical dendrites are of the CD1 pyramidal cells. We have the typical two-pathway experiment like Tom Adele was describing yesterday. So we do priming stimulation on one set of inputs, one set of axons, and then we look for how easy it is to get LTP or how much LTP you get on a second pathway in the same general dendritic region. And we can show that this is based on some earlier work from John Wagner, actually. So we just took his model of the preparation and got something very similar, where you can see that the priming activity reduces the amount of LTP with your different LTP protocols and increases the amount of LTD. So it's kind of BCM-like. So that was a good starting point. Okay, now we're going to figure out how it all works. So one of the first tests, we said, okay, if it's a cell-wide effect, then we should be able to prime anywhere in the cell and still affect LTP sort of anywhere else. And so the next thing we did was we did the priming stimulation up here in the basilar dendritic region. So we stimulated up here in stratum oriens and then did the LTP in stratum radiatum. And sure enough, LTP was inhibited and LTD was enhanced by stimulating on the other side of the cell body. And it still worked. So all going well for the BCM model. Now, without going through all the data, it started to fall apart at this point because we could block postsynaptic cell firing during the priming stimulation. We could actually block the depolarization, and we still got the priming effect just as well as if we didn't. So it actually is what we're studying here now is not the BCM effect or proposed effect, it's something else. And all the usual cast of characters in the kind of plasticity plays didn't seem to be involved during priming. Activation of MD receptors, activation of L-type voltage-dependent calcium channels, synaptic inhibition, so important, but not for this. But the one thing we found that did seem to be involved was release of calcium from intracellular stores from the endoplasmic reticulum. Via the IP3 being released and moving in and activating IP3 receptors on the stores, which proposed a certain conundrum here, because if we're giving a priming stimulation up here and there's calcium being released from intracellular stores in the basal dendrites, there's no evidence that I've ever been able to see that although there's a kind of a calcium signaling pathway within dendritic regions, there's no evidence that I've been ever anyone to show that it actually can traverse the cell body. So whatever calcium is being released here, it's not going to be getting over into the apical dendrites. So how is the communication actually happening then, is the question. And so we started thinking laterally, like there's not just neurons in the tissue, there's lots of other cell types as well. And I'm showing you some labeled astrocytes, for example. This is labeled with sulforhodamine 101, or you can do immuno with GFAP, and you can see that astrocytes are in red here everywhere, in CA1 and dentate gyrus, I mean, basically in the whole brain. And so it occurred to us that maybe astrocytes were providing this long-range signaling mechanism. So we had this working hypothesis that they sense neural activity. Actually, this is well known. We know that they sense neural activity, but they then operate to adjust the thresholds for future synaptic plasticity in the network. So I've got a new term called network metaplasticity. And like I say, to us it made sense once we got rid of thinking about just neurons, because first of all, astrocytes express a lot of receptors that couple to IP3 signaling, inositol triphosphate, binding to receptors in the ER and releasing calcium. Glutamate ones, acetylcholine ones, ATP ones, and so forth. Furthermore, these astrocytes talk to each other. Okay, so they have gap junctions for molecules to go through. They have hemichannels or other release mechanisms for molecules to leave, and then they can move and diffuse and activate other astrocytes. So they're very capable of talking to each other. And finally, they release glial transmitters. You might recognize glutamate. Well, not just neurons release glutamate, so can astrocytes, for example. And so these three are ones that I'm going to be kind of focusing on in this talk. So they can talk back to neurons by the glial transmitters that they release. Yo. To excitatory and inhibitory.
So do they know whether it's excitatory cell firing versus inhibitory cell firing in the same way?
I'm not aware of GABA receptors activating calcium responses in astrocytes. That's not to say they don't happen, but I'm not aware of that. They may well have GABA receptors. They do a lot of GABA transporting, but I think our effects, I think, are from excitatory transmitters. Okay, so then the question is, well, actually, do the astrocytes actually respond to the priming activity in radiatum when you stimulate in Oriens? And we had to go to Brisbane and Pankaj Shah's lab to do this, but we could see that all the astrocytes as you give repeated stimulation of the sort that we did for priming, you get these astrocyte calcium responses in the stratum radiatum astrocytes, and you can repeat it and you get it again. And virtually all the astrocytes in the zone that you can see, that can visualize, were responding. So in principle, astrocytes could be involved. That doesn't mean they are. It just means they're listening to what's going on, but are they actually doing anything? And there's various ways to approach this, but I'm going to show you what I think is the clincher for the deal. And the clincher for the deal was to patch a single astrocyte in stratum radiatum. And for those you know, of course, the astrocytes are not excitable. They don't fire action potentials, whatever. So they have a low membrane resistance, and they basically have, no voltage, obvious voltage-gated conductances, at least in this voltage range. But the interesting thing is, because they are low resistance, you can actually record the local field potential around that patched astrocyte, okay, as you can see here. And you can actually, so you don't need to have an electrode sitting outside in the extracellular space. You can record the field through your patch electrode on the astrocyte. So what we decided to do is to dialyze the cell with EGTA, which is basically a sponge that soaks up calcium. So anything that gets released from stores, or maybe even comes in from the outside, gets soaked up by the sponge, and then it can't work. Now, the first thing you learn, which we should have known, but we had to learn for ourselves again, was that as soon as you put EGTA in the astrocyte, it completely blocks LTP by itself. And that's because it prevents the astrocyte from providing one of the secret sauces you need for LTP, which is serine, which is a coagonist for the NMDA receptor. So in order to get LTP back, so we could actually find out if we can inhibit LTP, we have to bathe the slice in serine, D-serine, so that we get the LTP back in our clamped, what we call calcium clamped astrocyte. So here it is in red. This is the LTP measured through the membrane with sort of control LTP. And then there's a priming effect in just normal astrocytes. You can see it's reduced. And then in the calcium-clamped astrocyte, the LTP is back to normal. So this astrocyte was critical for the priming effect in the local region that the astrocyte is communicating with. We did a couple other experiments just to confirm that. So instead of dialyzing with EGTA, we dialyzed with heparin, which is it binds IP3. So it's like a sponge for IP3 now. So if IP3 is being released at the membrane and moving to the ER, it gets taken up by heparin. And the exact same thing happens. We get normal LTP in red. The priming effect occurs if there's no heparin. If you have heparin in that astrocyte, LTP is blocked. The priming effect is blocked and LTP is restored to normal levels. And then finally, we got in some IP3 receptor 2 knockout mice. So the IP3 receptor in astrocytes, that is, it's pretty specific for astrocytes, is the receptor 2, IP3 receptor 2. If we have that knocked out in wild-type mice, we get the normal priming effect, but there's no priming effect in the IP3R2 knockout mice. So we think some of this, plus some other data we have, strongly indicates that astrocytes are critical at some point, at some stage of this long-range communication to produce this priming effect. One of the molecules we think is involved, signaling molecules somewhere in this signaling system, is tumor necrosis factor alpha, a cytokine. Pro-inflammatory or anti-inflammatory, depending on the levels and the duration that it is around. But we showed that if you bathe the slices in a TNF-alpha antibody, you lose the priming effect, goes from red to green. And TNF protein, if you just bathe the slices with it for a while and then wash it out, then that itself can produce the priming effect and inhibit LTP. And this is acting, there are two receptors, basically, TNF-R1 and TNF-R2. TNF-R1, for various reasons in the literature, was our target as most likely to be involved. And indeed, again, we got some knockout mice, and the wild-type mice show normal priming effect, and the TNF-R1 mice, the knockout mice, don't show the priming effect, basically. Yes?
Sorry, from the previous result, how do you bring together the fact that you have the taxon result for interfering with one astrocyte versus interfering with the whole network in the IP3 receptor lines?
I have no problem with putting those together. The idea is that the astrocytic network is being activated. Basically, all of them are being activated. So the synapses that are near to each of those astrocytes can be affected. With the single astrocyte calcium clamp, we're only looking at the synapses around the astrocyte because we're recording the local field potential just through the patch pipette. So, and maybe actually with the calcium clamp, maybe a few other astrocytes around are getting the EGT as well. So there is a zone of control. But between single astrocytes and the whole body of astrocytes manipulating them, we're getting the same effect, which you might expect because we're thinking the astrocytes are communicating through the whole network. Does that make sense? Okay, and then the next step, and maybe the final step in the process, is to signal back to neurons. And we think that this is, speaking of extrasynaptic receptors, we think that this is going through release of glutamate from the astrocytes, which as people have shown in other studies that TNF can cause glutamate release from astrocytes. And because the astrocyte processes are not really in the synapses themselves, but are just around at the zones, the edges of the synapses, that the glutamate binds to extrasynaptic receptors, particularly gluN2B containing receptors, which we know can impair LTP when activated. And indeed, if we put in a drug that blocks gluN2B containing NMDA receptors, we block the priming effect either from electrical priming or from TNF priming of the synapses. Now, so that was enough surprise, but then we kept being hit with more surprises. So actually, this is not a cell-wide effect, as it turns out, because when we started looking at LTP and other dendritic zones, so we have these three, the basal dendrite, stratum oriens, stratum radiatum, and stratum lacunosus and locularia we've been hearing about with the entorhinal cortex inputs. We do electrical priming in stratum oriens. The only priming effect we see is in this dendritic zone in the apical dendrites. A second pathway, even in the same region where we did the priming, the LTP is fine. And it's also fine in SLM. And when we get basically the same result with applying TNF as the protein as a priming signal, the stratum radiatum is particularly sensitive to the application of TNF in terms of inhibiting LTP. SO and SLM didn't respond. Although, to be fair, if you give enough TNF, then you can inhibit LTP in these other zones. But with electrical priming anyway, it seemed to be a reasonably specific effect for stratum radiatum. So we have this system, quite unfortunately a complicated diagram. I don't expect you to work through it, but we have this idea that, and there's some things that we haven't talked about, I'm sure you're going to ask, but there's various molecules released that can activate astrocytes. They communicate with each other, ultimately release, including through TNF probably, they release glutamate where we think it binds to extrasynaptic gluN2Bs, and that inhibits stratum radiatum LTP. Now, of course, there are other glial cells there in the mix, and that includes microglia. They can also be activated by synaptic activity. They also can communicate with astrocytes. They also release TNF, and we're in the middle now of trying to figure out whether microglia are actually play a role in this whole cascade of effects. But wait, there's more. So astrocytes, as I said, are everywhere, right? So they're all through CA1, but they're all through dentate gyrus and CA3 and everywhere. So I had this crazy idea, unfortunately my student took up, it's like, well, does the priming effect actually cross the hippocampal fissure? Seems unlikely. Why? And affect LTP in the dentate gyrus. Now you can see that there's quite a plexus of astrocytic processes around the hippocampal fissure because of all the blood vessels that run through there. So it's not completely implausible there might be astrocytic communication across the fissure. And there have been a few studies, a couple studies suggesting this could happen. And sure enough, when Shruti Satish does these experiments for a PhD, she did either a high-frequency priming stimulation or a theta burst priming stimulation. In both cases, LTP was inhibited in the middle molecular layer of the dentate gyrus. So not only is this soup or whatever is happening occurring in CA1, but the communication also goes to the dentate gyrus. And we also did it with, so I should have said, these are 30-minute delays between the priming stimulation and the LTP induction. This effect generally lasts only 60 to 90 minutes, so these aren't really long-term effects, the metaplasticity. But if you do shorter, like 5-minute delays, you get a very strong priming effect. And then when we moved in vivo to anesthetized rats, and we did the same experiment, priming in stratum oriens and doing LTP in the middle molecular layer of the dentate gyrus, we also got impaired LTP. So it's not just a slice effect. And are astrocytes involved? Yes, but again, what's the secret sauce for LTP in the dentate gyrus? Well, it's not D-serine, it's actually glycine, which is another coagonist with NMDA receptor. So you have the slices bathed in glycine, and then we do the calcium clamp with the single astrocytes in the dentate gyrus, and we get the impairment of the priming effect, as you can see here, with the calcium clamp. And we got the IP3/2 receptor mice, knockout mice, and they also had no priming effect in the dentate gyrus either, compared to the wild types. So we have this rather strange set of effects that I'm hoping you guys will all be able to help me understand, wrap my mind around. So we have a priming effect in SO, and quite possibly the same is true with SR, has no effect on LTP, it seems, on at least the way we've done the experiments in the same layer. It inhibits LTP in this layer, does not inhibit LTP in this layer, and inhibits LTP in this way. Now, the hippocampal circuitry is such that, the dentate gyrus projects to CA3, which we haven't studied, which projects to both stratum oriens and stratum radiatum. So this is one of the things that confuses me. Why, for the same input, is one of them affected and one of them not, depending on which dendritic compartment you're doing. But the really interesting thing and novel thing for our perspective is that there's this new backwards communication from CA1 back into the dentate gyrus via the astrocytic network controlling plasticity. And as I say, I think the stratum radiatum activity can do the same, although we haven't tested this one into dentate gyrus. And who knows, maybe activity in SLM can do the same, that we haven't done that test either. So we've had these various forms of metaplasticity that I described at the start. So now I'm adding a new one, which I'm calling network-wide, although it's obviously not the entire network, but parts of the network, network-wide metaplasticity. And maybe in artificial neural networks, this might be like changing the learning rate kind of plasticity in models for either all of the networks or parts of networks, as it happens in deep networks. So to kind of conclude for astrocytes, first of all, in case you weren't aware, there's an online regulation of LTP via D-serine and glycine. But then that can also contribute a network-level metaplasticity across space and time, the whole space-time continuum, through the hippocampus via TNF signaling. Is this about homeostasis? I actually don't think it has anything to do with homeostasis, because it's so specific to dendritic compartments, and it's not the dendritic compartment that has the LTP or the priming effect in the first place. So it probably has something to do with biasing activity, kind of like what you were saying with your metaplasticity, biasing storage versus retrieval pathways in the hippocampus. And just to expand on that a little bit, first of all, rapid BCM-like metaplasticity from a modeling point of view may help stabilize synaptic drive following LTP events, but that's not what my story is about. My story is about there's a different form of metaplasticity that exists via activation of the astrocytic network. It's pathway specific, interestingly, and it even extends to a different subregion, revealing this novel backward communication from CA1 to dentate. And for those of you who know about synaptic tag and capture, this effect is in the completely opposite direction to what you would predict from synaptic tag and capture. I can expand on that if everyone is desperate to know what that's about. So in normal tissue, what does this mean? I don't know. We haven't done any behavioral experiments. We don't know. But it could be a way of stopping further plasticity at synapses where there's been plasticity so that it prevents interference from new learning for some period of time while consolidation is taking place. That's about the best I can come up with. But it is interesting to note that these IP3R2 knockout mice have impaired LTD and impaired remote memory, as if there may be some impairment of consolidation. And I could give you another story, but this mechanism can be aberrantly engaged in neuroinflammatory disorders, like in AD models, and it appears to contribute to the impairments of LTP and cognition in these models via basically having too much TNF around. So that's my story. I want to thank my series of PhDs and postdocs and my collaborators and funding agencies. Ju Chen provided the IP3R2 knockout mice. Thank you very much, and thank you for your attention.
Thanks. First of all, I think it's pretty wild that in the dentate gyrus, glycine itself is the glycine site agonist instead of D-serine. Is that because the astrocytes don't express serine racemase? Or why would there be a difference in the gliotransmitter for the glycine site?
I don't know is the answer. The sensitivity to serine and glycine seems to depend on the NR2 subunit composition. And I think there's some, my postdoc knows more about this, I'm afraid, but I think there's a difference in the subunit compositions at the synapses with NR2A versus, or GluN2A versus GluN2B, and I think that's one of the drivers for the relative sensitivities. I'm not saying that serine can't do it with enough, but they're more sensitive to glycine. Glycine can also do it in CA1 if you give enough, but there's a relative sensitivities.
Also, I mean, how do you think it might work mechanistically? Is this just kind of a passive property of gliotransmission where they kind of run out of, D-serine or glycine, and so you kind of get metaplasticity or a decrease in learning rate naturally? Or do you think it's more actively shaped by astrocytic processes?
Well, I'm not entirely sure what you mean, but I don't think the serine is being released by the tetanization. I think because it's there in the tissue. So I think because they've been patched for some time before we're producing LTP. So I suspect, although we've never measured it, that the serine or glycine is going down because of some ongoing release as opposed to stimulated release. Now, there's some debate about whether the astrocytes release D-serine, but there's actually some evidence now that it actually releases L-serine, which gets into neurons, which convert it to D-serine, which then get released again. So there is a communication pathway there, if that answers your question. Yeah.
Astrocytes in CA1 were shown to be spatially selective. Would you predict that the spatial selectivity of astrocytes is anti-correlated with the nearby neurons because they reduce they increase the region of LTD?
I'm sorry, I'm not sure what you mean by the spatial selectivity. I mean, astrocytes, they basically tessellate.
When the animal runs in the environment, the activation of astrocytes was shown to be selective for specific locations.
I see. Yeah, I don't know enough to really be able to comment on what to predict based on, let's say, environment experience on differential astrocyte responses and what will happen there. But one could imagine that if some astrocytes are strongly activated and other ones are not, then the synapses around those astrocytes will be from many neurons, not just one. But that conglomeration of synapses might be locally affected, and this might be part and parcel conceptually with the sort of clustered plasticity hypothesis, that maybe some areas, not only is there clustered plasticity in some areas, but it may be prevented from occurring in other areas through astrocytic zones of control.
I'm just trying to wrap my head around how this might function in vivo. So I don't know how many astrocytes you think would be involved in this process. Let's say during, for example, the establishment of a place field. And how would that, what's the range of that during some naturalistic experience? I think the previous question was kind of referring back to that. But it's the interaction with the dentate that, what's the advantage of coordinating broadly? Is it really regional? Because it can communicate to the dentate. Well, then you might imagine it can communicate to lots of other, nearby inputs in multiple neurons in CA1 as well. Where's the specificity?
I don't know is the answer. And maybe the specificity is not in the astrocytes, but in the synapses they're talking to. Because basically what I've shown you is that synapses have different differential sensitivity to this effect. So the astrocytes might be doing the same thing everywhere. I'm not saying they do, because could be kind of selective activation of astrocytes potentially. But it might be who they're talking to is what's providing specificity.
So they're sort of permissive, but you need some convergent postsynaptic activity plus the activation of these astrocytes.
Yes, well, I think the astrocytes are being activated by neural activity, so they do go hand in hand. And we think cholinergic input as well as glutamatergic input might be doing this. So they do go hand in hand, and then, the metaplasticity is not a direct thing. They're not causing the synapses to strengthen, they're not causing them to weaken, they're just changing their state so that when they try to have plasticity later on, the LTP's paired LTD is enhanced. So it is close communication between the cells. The synapses vary. And what really intrigues me is that the CA3 synapses in radiatum are different from the CA3 synapses in ORNs. Yeah. And so it matters what those synapses are.
I'm sorry, I think you showed with Pankaj Shah, you had imaged astrocytic activation, but I didn't totally absorb that. It looked like there were lots of astrocytes that were being activated. This was with a high frequency stimulus.
Yeah.
But perhaps in vivo with some more naturalistic experience, it would only be a small number of astrocytes that are activated. Did you look at that?
No, we haven't looked at that. And that's a very good question. I mean, obviously we're amping the system by high frequency electrical synchronous activation. So this is something that the network is capable of. Big question is, how does it work in vivo situations?
Oh, very interesting. Thanks. I was wondering if this mechanism is related to astrocyte-dependent short-term plasticity?
Well, there certainly, I mean, if you're talking about the kind of transient heterosynaptic depression that astrocytes seem to mediate, so you give high-frequency activity. I think Tom was showing some transient depression on another pathway. That is astrocyte mediated. It's probably involving adenosine, but maybe other things. But it is only short term. And as I mentioned, if you go back, we look at those slides, the priming activity has basically no effect on basal transmission in the other pathways that are being studied. So I don't think the long-term effect that I'm describing requires that heterosynaptic depression. It can happen sometimes, but I don't think it's a driver for the long-term metaplasticity change.
Okay.
Okay, great. I was just looking at your point number 4 about this is opposite synaptic tag and capture, and I was wondering, is there something in your data that's not compatible with there being a cell-wide effect that's countered at the apical dendrites by a tag and capture? Like, could it be that there are two things that add up to nothing, no change in the LTP capacity at the synapses where you primed, but a decrease in some other places?
So for those who don't know, synaptic tag and capture just means that if some synapses are activated and they sufficiently strongly to generate protein synthesis, then other synapses stimulated at a different time can, even if they're weakly stimulated, can capture the proteins that are made from the strongly stimulated synapses and use that to build late phase LTP. So that's a good point in the sense that in ORNs particularly, there may be sharing of material and sort of obscuring or preventing the inhibition of LTP because there's some positive thing going on. But I don't know how it would work in SLM. I don't see it traveling.
But if you block TNF, then do you see the enhanced LTP in ORNs? If you block your TNF effect,
in the TNF,
then you might, if there are two things happening, you might see enhanced.
Yeah, I don't think we see any significant alteration in LTP in the TNF knockout or R1 knockout mice, but it's a good question. Yeah.
Okay, great. So we are right on time. Let's go for coffee and be back at 11 for the next talk. And let's thank Cliff and Richard again.